All right, good morning, everyone. Today is August 5th, 2021, and today we're going to be talking about microorganisms as vectors for disease, in addition to a couple different models for disease transmission. We're also going to go over viruses because for the most part, we've been learning about bacteria. Part of the reason for this is that you can grow bacteria into colonies, but you can't really do the same thing with viruses because they're much, much smaller than bacteria. So a disease caused by microorganisms usually refers to a disruption of homeostasis because it impairs healthy functioning of parts of your body or your cells. This could be on the level of an organ like your lungs, or specifically how your cells will process different things. Like when a virus enters your cell, it will hijack different machinery inside of your cell to make copies of itself. Different diseases can be attributed to aging, like Alzheimer's. You can also have genetic diseases like autism, which also has environmental components. In addition, diabetes also has environmental components, but for the most part, when we think about diseases, we think about those caused by bacteria or viruses. And all of these infectious agents that we've been talking about are called pathogens. And you can break down pathogens or different diseases into communicable or contagious diseases and those which are non-communicable diseases. The communicable ones or the contagious ones are those which can easily be transmitted from one person to another. You can think about the coronavirus and how quickly that took over our planet to spread in almost every single country to recently has infected 200 million people, and those are just cases which have been accounted for. On the other hand, non-communicable diseases are chronic diseases which cannot be transmitted from one person to another. We just talked about briefly autism and diabetes. These are genetic or environmental and cannot be passed from one person to another. There are different modes in which you can acquire infectious diseases. You can acquire them through direct transmission or indirect transmission. Direct transmission occurs when you contact an infected individual. Examples of contact include kissing or having sexual intercourse with them. In addition, when a person speaks, you can sometimes see respiratory droplets flying out of their mouth. Respiratory droplets are one of the main forms in which we know that the coronavirus spreads. In addition to direct transmission, indirect transmission occurs when we have an infectious agent that gets transferred from one area to another without so-called direct contact. You can have airborne transmission, which is sort of similar to respiratory droplets, but the difference between respiratory droplets and airborne transmission is that airborne occurs through droplets, which are so small that they become into the form of an aerosol, and they sort of stay in the air, and if you breathe them in, you can become infected with a virus like the coronavirus or the flu. Fomites are inanimate objects. Think like a door handle or countertop. If you touch this surface and then afterwards you touch your face, your eyes, your nose, your mouth, You can become infected with a virus like this or another infectious pathogen. However, this particular mode of transmission is not really well recognized, especially not with coronavirus. In addition, there's also vectors. Think about like animal vectors or mosquito vectors. For example, mosquitoes are able to transmit diseases like West Nile virus, Zika virus, or dengue virus. In general, when we look at directly transmitted diseases, these are the ones that are transmitted through respiratory contact or direct contact. We see that the directly transmitted ones over here on in the right side of the graph have a very low mortality rate. However, when there are vector-borne diseases like Zika virus, dengue, West Nile, we see that the range of deaths is actually higher. And this is because directly transmitted viruses will require a relatively healthy host to be able to spread the disease, whereas vector-borne diseases will rely on something other than the human to spread the diseases. And because you don't have that necessary requirement for direct contact, you can have a much greater mortality rate. On the right, we have a graph comparing two types of cholera. Cholera has caused, I believe, 7 pandemics, and the reason why classical Vibrio cholera is so deadly is because typically it's transmitted through infected water. And when you have a larger fraction of the outbreaks that are waterborne, you will also have a higher percentage of deaths per the infection. This is because when the infection is waterborne, it does not require a healthy host to spread. So because we don't have this direct transmission, there is no necessity for human contact. And this just makes in this case, cholera is a bacteria. Sorry if that wasn't clear. This just makes this bacteria much more deadly because it doesn't require any direct contact. We're going to talk about why that is in a little bit. Now, there are different sources of disease, which we call reservoirs. This refers to the place where a microorganism normally lives, where it grows, and where it multiplies. Humans can be a reservoir, for example, for coronavirus. Animals can be a reservoir for all sorts of diseases, including diseases which have not yet entered the human population, because animals have their whole set of different viruses, which, depending on the similarity, for example, between entry in humans and animals, diseases, viruses can mainly jump from animals, especially mammals, to humans. You can also have a soil reservoir, for example, in the bacteria Clostridium botulinum, that one primarily lives in the soil. Oops. So you have an infectious agent. This is the pathogen that we talked about that in most cases will make people sick. The reservoir is where it lives. For example, if you are sick with the flu, because you're sick, you sort of become a reservoir for this virus. And the way that the virus will leave your body to infect more people is when you sneeze or cough, or when you cover your cough with your hand and then you touch another surface. That's an example of indirect transmission. Once the germ is out, if it's in the air, it can spread to other people via aerosols. or via indirect contact. Next, the virus in this case has to find its way into another person. If it's airborne, that person will breathe in the virus and eventually cause this person to become sick if they are susceptible. Not every person that gets exposed to a virus is susceptible to severe disease. And we've seen this with coronavirus, right? We've seen that some people are just predisposed to having a much more severe infection, usually because of pre-existing conditions like diabetes, hypertension, cardiovascular disorders, um, immune system conditions, autoimmune conditions. Animals have lived with people since the beginning of agriculture. Animals can provide us with food. They can provide us with dairy products. Some people keep animals as pets. In addition, animals have also been used for sport, like horseback riding or things like hunting. And because of this, because we've gotten so close to animals, animals can transfer their viruses to us, and this is called a zoonotic disease or a zoonoses. We can have different viruses, for example, the flu. Most flus that we have every year, those are avian flus. They come from mostly chickens. I believe in 2009 or 2010, there was an outbreak of swine flu. Some people believe that the coronavirus came from either a bat or a pangolin or a civet, after which point it jumped into an intermediate host before it entered the human population. In addition, the same way that people have viruses, we can also transfer them into animals and make them sick like that. Because we've gotten so close to all these animals, it is estimated that approximately 60 to 75% of all new emerging infectious diseases are of zoonotic origin, which just makes it incredibly important to study where these outbreaks come from and also how they will affect our daily lives. Intro to epidemiology. This is very brief. I just wanted to go over it because your HOL talks about it for the first for the first experiment. You have to fill out a table basically with the incidence of different diseases like chlamydia, maybe Zika virus, and other pathogens that are present in the United States. So incidence refers to the new cases of a disease within a population at a specified time interval. And prevalence is the proportion of people within a population who have a particular disease or attribute within that defined period of time. The difference between these two, and I know they sound very similar, I just wanted to point out the difference, is that incidence only refers to new cases, whereas prevalence refers to both new and pre-existing cases in that defined period of time. Briefly wanted to go over what viruses are. Viruses are obligate intracellular pathogens. This means that they absolutely have to go into your cell, use the machinery inside of your cells to make more copies of themselves. And this is because viruses lack the machinery to reproduce by themselves. In general, viruses consist of a genome. The genome can be RNA, single or double-stranded, or DNA, single or double-stranded DNA. This one here, I believe, is an example of the coronavirus. Actually, the coronavirus doesn't have a capsid, but almost all viruses have a lipid envelope on the outside. And this is important because the lipid envelope will merge with our cells. And embedded within the lipid envelope are spike glycoproteins, specifically in the coronavirus. And these spike glycoproteins interact with our cells receptors on our cell surfaces so that they can become internalized. Where do viruses come from? There are three general hypotheses for the origins of viruses: the regressive hypothesis, the progressive hypothesis, and the coevolution hypothesis. With the regressive hypothesis, it is predicted that viruses are degenerate forms of intracellular parasites. You can think about them as once having been a little bit more complex organism, like a bacteria, which lost a bunch of genes, and over time, because they lost the ability to do things like replicate themselves, they adopted a parasitic lifestyle. There's also the progressive hypothesis that states virus fragments of genetic material like RNA escape from cells. These are normal pieces of DNA or RNA that somehow gain the ability to make copies of themselves autonomously and after many years to evolve. It is believed that DNA viruses arose from plasmids or transposable elements, after which point they acquired coat proteins and the ability to be transmitted. Retroviruses, such as HIV, are thought to have derived from retrotransposons, and RNA viruses from mRNA. There is also the coevolution hypothesis, which states that viruses co-evolved with life, that their evolution might go all the way back to the RNA world. The RNA world is thought to precede the existence of DNA. Now, all three of these are just hypotheses. It may be that none of them are true or all of them are true. Briefly, virulence versus pathogenicity. Virulence is the harm done to a host from a pathogen. It varies greatly between organisms. For example, cholera can be lethal. On the other hand, cold viruses produce generally a very mild infection. On the other hand, pathogenicity refers to whether or not the organism can cause disease. This is an evolutionary biologist's point of view of virulence. It states that, and this is pretty logical if you think about it, new associations between a virus and its host lead to virulence. And it's thought that a virus should not harm its host because it requires it to survive. There are three hypotheses that explain the evolution of virulence. They are the short-sighted hypothesis, the coincidental evolution hypothesis, and the trade-off hypothesis. The short-sighted hypothesis states that pathogens to stop reproducing once the population or the number of copies of virus reached is at the point of harming the host. Myopia or short-sightedness is a premise for the theory of natural selection. However, natural selection is not controlled by viruses, just sort of happens. Mutations occur. If those mutations are successful, they will be kept. If not, it will keep mutating to the point where it gains a mutation that helps it. We're seeing this right now with different variants like the Delta variant. There are multiple mutations in the part of the virus spike protein, which binds to the receptors on our cells. This is incredibly important because the Delta variant is actually much more transmissible, and we're going to talk about why that might be with the trade-off hypothesis. This of the three of them is considered to be the weakest hypothesis because it underestimates the power of natural selection. Like it says earlier, it's not an intelligently controlled process because natural selection can evolve solutions to problems which can resemble foresightedness. There is the coincidental evolution hypothesis that states virulence in humans may not be a target of selection itself, but rather an accidental byproduct of selection in other habitats. For example, Clostridium tetani or the tetanus bacteria produces a neurotoxin that is lethal to humans. However, it cannot be transferred in between humans. You may be familiar with tetanus. You are required to have a tetanus shot before you start college. This toxin does not particularly do anything in the benefit of the bacteria to humans. However, it is a plus for the bacteria in the soil. It probably acts something like an antibiotic to kill off other bacteria. This hypothesis of evolution states that virulence in humans is coincidental. However, the drawback to this idea is that it doesn't explain most cases of virulence. The trade-off hypothesis suggests that natural selection should strike an optimal balance between the costs and the benefits of harming hosts. There is a trade-off between the rate of transmission and the duration of infection. If you kill the host, the human that a virus infects, this reduces transmission and also reduces the duration of infection. In addition, if you have a virus that makes people really sick and the host is bedridden, this person's probably not going to get out of bed. It probably can't infect a lot of people. However, if you have a virus that's less virulent to the point where in some cases it's like coronavirus, it produces very mild symptoms or it's asymptomatic, this allows for more chance for transmission because the person may not even know that they're sick. It might not really affect them to the point where they have to stay in bed all day. With the more virulent example, at most the people that would get infected are the people who are immediately taking care of this person. And the trade-off hypothesis suggests that there is a fine line between the success of transmission and the mortality of the host. In this hypothesis, the virulence of a pathogen may increase if it sufficiently increases the pathogen's chance of being transmitted from one person to another. Coronavirus, we know that it can be spread through both airborne transmission, which is considered indirect, and respiratory droplets, which is direct. Some people will experience severe disease while others are asymptomatic. Going back to the trade-off hypothesis, there is the trade-off between the mortality of the host and the success of transmission, depending on how virulent the virus is. There is a principle called R-naught numbers, which is a measure of how infectious or how contagious an infectious disease is, and it also tells you on average how many people one individual can infect. If the R-naught value is less than 1, one person will on average infect less than one other person. Typically means that the infection rates are under control. If the R naught equals one person will infect exactly one other person. If the R naught is greater than the one person will infect severely more than one person, and at this point, the infection spreads rapidly. This is a graph of cases in New York State, I believe, or New York City. Excuse me for not knowing. However, you can see that at the beginning to middle of March, the cases were increasing very, very quickly. And at this point, the R naught value is much greater than 1 because we see very high transmission in the community. This is when New York State was blindsided by COVID. And then over the summer, in the summer of 2020, we see that the R naught was much less than 0 because the infection rates were under control. And one person in general infected less than one person on average. Briefly wanted to talk about the different kinds of vaccines because they're very, very popular right now. Some of the earliest vaccines were subunit vaccines. Actually, the earliest vaccine goes back to, I believe, the end of the 17th century, around 1795, when Edward Jenner took a piece of the smallpox virus. Yes, the smallpox virus. And he hypothesized that if you expose a small piece of the virus to an uninfected individual, that this person will develop immunity. Subunit vaccines typically use proteins nowadays. We can grow the proteins. We talked about cloning. We can grow the protein in a bacteria, isolate it, and then use it to create a vaccine. There are also inactivated vaccines, which are whole virus vaccines. In general, the virus is killed. This is the flu vaccine that you probably get every year, in addition to the polio vaccine and any rabies vaccines. There is also another form of the flu vaccine. It's called a live attenuated vaccine, which has a weakened virus. However, that one's not as available. I've never gotten it personally. The live attenuated virus vaccine is, I've heard that it's like an intranasal spray. There are virus-like particles, which will be very similar to the actual virus itself, but still cause immunity for it. And some of the coronavirus vaccines are made with viral vectors. For example, the Johnson & Johnson vaccine uses DNA inside of a adenovirus vector. The DNA will be transcribed into mRNA, and the mRNA will be made into protein. And then your body will create antibodies against that protein after it arrives at the cell surface. In addition to Johnson & Johnson, the AstraZeneca vaccine also uses a viral vector. It's also an adenovirus. And also the Russian vaccine called Gamaleya. Now, the Pfizer and the Moderna vaccines are created with mRNA technology. They use the mRNA from the coronavirus spike protein. It's just the spike protein inside of a lipid envelope. The lipid envelope helps to keep it stable inside of the body because mRNA can easily degrade. by injecting an RNA vaccine into someone. This gives your body the instructions, just like with the Johnson & Johnson vaccine, to create the vaccine itself. You're creating the protein, which your body will make antibodies against. Because of all the new variants, I highly recommend vaccination. Typically, the way that vaccines are tested for efficacy is clinical trials will be divided into two groups. There's phase 1, 2, and 3. However, for the coronavirus, phase 1 and 2 were combined into one, and phase 2 and 3 were combined into another trial just to get around the bureaucracy of the approval at the FDA. However, you should also know, if you're kind of skeptical about the vaccine, is that these clinical trials enrolled about twice the number of people on average compared to regular clinical trials. So in total, both the Pfizer and Moderna clinical trials enrolled about 40,000 people each. So half of those people receive a placebo, which is in most cases, it's compared to a sugar pill. However, it's just like a controlled treatment. And the other half receive the mRNA vaccine. The control group is just to test whether the mRNA vaccine is really effective. It was found that most people that had the coronavirus that participated in the clinical trial were from the unvaccinated group, or those that received the placebo. Bottom line, the vaccines are safe, they are effective, they are being provided for free. That's not the case in other countries. You have to pay a lot of money to get these vaccines, so we're really lucky to have them. So even though these vaccines use brand new technology, mRNA vaccines have been proposed for almost 20 years. They have been studied in animal models like mice, chimpanzees. So if you haven't gotten your coronavirus vaccine, I highly recommend getting it, because there are many other viruses which were completely eradicated with vaccines, like polio. We don't really think about polio because nowadays it has been completely eradicated, but in the 1950s, polio would cause paralysis of some people. It would cause death in some people, and some children even had to suffer through being placed inside of an iron lung. In addition to polio, we've also eradicated smallpox, measles, mumps. We're able to control flu every year because a lot of people get vaccinated. So bottom line, vaccines work. Vaccines take a small piece of the virus to present it to your immune system. The reason for this is that viruses are not living. They don't have ribosomes. They can't make copies of themselves. When you think back to all of our other lectures on bacteria, we learned that antibiotics target specific parts of the bacteria, like ribosomes or cell wall synthesis. However, because viruses don't have these components, we have to take a small piece of the virus to present it to our immune systems so that we can get immunity without causing disease. Okay. There is another exercise on HOL. You have to take the sodium hydroxide from your kits, pour it into a cup. In addition, have about 9 other cups on your table. Fill them up with water, and then You're going to take the sodium hydroxide cup after you're going to have an assistant take the sodium hydroxide cup and then kind of just pour liquid in and out of all the different cups. Your assistant isn't supposed to know which cup has sodium hydroxide in it. This is supposed to be like a simulation of people sharing different drinks or food when they are together. And after your assistant has, like mixed all the cups, all the liquid in the cups, you're gonna take the other part of the experiment. There is a small dropper called phenolphthalein. Phenolphthalein is a pH indicator. So at a pH of 8, it will turn pink. So you'll see that like in the cups that have sodium hydroxide in them, they will turn a slight shade of pink, and this just means that in theory, like this model that we're running, that the virus has spread in our model.